Hello everyone, I'm Shannon from UC Berkeley. I'm delighted to share with you today highlights of my research from single molecule biophysics to live cell force dynamics. I've studied the processive ribosome translation, the folding of the nascent chain during their synthesis, the remodeling and scission of plasma membrane for vesicle budding, and the propagation of membrane tension upon cell protrusions. The common theme, or the very nature if you will, is that mechanical work is at the heart of these dynamic processes so as to carry out the required physical task. Therefore, I can apply and measure forces using optotweezers adapt for each biological system and resolve their underlying dynamics. Ultimately, I want to expand the scope of my work to explore cellular force dynamics in situ, such as chromosome segregation during mitosis, and investigate biomechanics across scales. In my PhD, I resolve how the ribosome can be programmed to slip and switch mRNA reading frame during translation. And that slippage so minute in size can be characterized by optotweezers and manifest as dynamic excursion during the forward translocation step by the ribosome. This frame-shifting scheme promoted by specific mRNA elements is often used by bacteria and heavily exploited by viruses such as HIV-1 and SARS-CoV-2 to translate multiple proteins from a single transcript. When I used mass spec to examine the translated product, I found that ribosome can in fact slip backward by one nucleotide and by -4 or +2, all entering the mRNA -1 frame, rendering polypeptides one residue longer or shorter. And these species were never resolved before. I also found incomplete polypeptides terminated upon ribosome slippage, indicating that translation fidelity check on base pair mismatch is still at play on the ribosome post-translocation. During postdoc, my colleague and I used optotweezers to resolve how a nascent transcript of the signal recognition particle RNA evolves through sequential folds as it gradually emerges on the RNA polymerase. Using in vivo assays, I found that the formation of a non-native obligatory intermediate unexpectedly permits co-transcriptional RNA processing and remove its precursor fragment. However, overstabilizing this intermediate will impede folding transitions to its final native fold, causing a fatal loss of function that impacts cell viability. Therefore, the biogenesis of this essential RNA is modulated co-transcriptionally by its own folding. This work is a stepping stone to my current K99 and future grants to study co-translation protein folding. I will investigate the fold switching of metamorphic proteins, like MET2, and identify where along its folding trajectory lies the vivification point for the protein to switch between its functional folds. For increasingly complex systems, I adapt optotweezers and, for example, study the ATP-dependent force generation by Ascor complex, which sever the membrane neck of an outward-budding vesicle. We successfully recapitulate this dynamic process using reconstitute vesicles, which encapsulate all the active ingredients. Then UV uncage ATP to initiate membrane remodeling. And concurrently, we monitor the rise in force along the membrane tether held by a bead in an opto trap. In another project, I integrate our tweezers with optogenetic control to trigger localized cell protrusions. Using a dual trap setup, we reveal that actin-driven protrusions elicit rapid membrane tension propagation across the cell, and demonstrate that membrane tension can serve as a long-range integrator of cell polarity during migration. My effort in innovating tools like integrative optical trapping is motivated by the scientific community to study biomechanics. And it is a manifestation of my own desire to directly measure and broadly explore the mechanical aspects inside and around cells. Narrative in the mechanical dimension, including force dynamics of spindle assembly modulated by MET2, which my group will study, contribute greatly to the biological interconnectivity and operation of living systems. However, this dimension remains largely invisible in the cell atlas resolved by modern imaging techniques. Therefore, my ultimate goal is to establish in situ force sensing fluorescence spectroscopy and microscopy with novel molecular probes and instrumentations. We will visualize the prevalence and magnitude of forces on a broad set of cellular complexes. And by incorporating them with our current knowledge space, we can animate live cells in 4D beyond phenological descriptions and open new fields of study in biomedical sciences. I very much look forward to working with you all. Thank you.